All right, here we go with another AP Biology review. Unit 6 is going to be split up into two sections. This one about DNA and protein synthesis, which is the main chunk. And then the next one will be about gene expression and biotechnology. The name of the game in life is making proteins. Proteins are the molecules that really carry out all of the functions of life. For example, if you want to taste something, you need these taste receptors that are sticking out of the ends of your cells, membrane-bound receptors, right, that are going to attach to the ligands, which would be the molecules in your food. And those, of course, then send signals through the communication pathway and all that stuff to your brain. Right, so if we want to live, we got to keep making all these different proteins, and the information for that is found in our DNA, right? And so we already talked some of this. There's another video about this, but we have our phosphate group and our deoxyribose sugar, right? And that's for DNA, of course. Then you have the nitrogenous base of the A's, G's, C's, and T's, right? So this whole thing is called a nucleotide. The nucleotides then get joined together into a polymer. This one is interesting, though, of course, because we have our double-sided, double-stranded molecule, where the base pairing rules, of course, I didn't even make a slide for base pairing rules. I'm pretty sure you know that the G's and the C's and the A's and the T's go together. Hopefully you remember that we hold these together with hydrogen bonds because we're going to break the sides apart quite often. And something that, again, I mentioned before when we talked about structure, but this is going to be the time where it starts to become actually be important that we realize that the strands go in different directions and that the directions have names, that the top of the molecule up here is the 5' end, and then down here is the 3' end. Right, and so we're always going to be looking at things going from from the 5 to the 3' direction, which again, we'll get back to that soon. Here's just a picture to show you a more detailed version, and you can see the 5' and the 3' where those numbers come from. Of course, we have the double helix. We actually read the original reporting from James Watson and Francis Crick about the double helix structure. And one of the things in that paper was the idea that once they figured out the structure of the DNA, they realized that the base pairing rules and the structure of DNA itself explain how you use it. Right, and so they were more concerned with copying mechanisms. They wanted to know how you copy your DNA. So we will take a minute here to talk about replication. This isn't for protein synthesis, but of course we just finished talking about some units where we need to make new cells. And of course replication is very similar to transcription that we'll get to in a minute. So of course, there's a big difference between prokaryotes and eukaryotes, and that being that the prokaryotic genome is just one circular chromosome, and it's significantly smaller than even our smallest chromosomes. So prokaryotes can start in one place and then just work their way in both directions until you get two copies of the same DNA, right? They can just start in one place and go from there all the way around the circle. But in a eukaryote, we have, as like for humans, we have 46 chromosomes, and each one of those chromosomes is super long. So we're going to start in lots of places called the origin of replication. And this is going to make the replication go in both directions. As you can see in the pictures, the arrows are going both ways, but eventually the bubbles are going to merge together. And so one particular molecule is not going to start at the beginning and get to the end. A lot of molecules are going to start, and they're going to all work together and make one little piece of the bigger copy. You probably hopefully you remember this in general, but the big thing here is the enzymes. You need to know that there's an enzyme called helicase that opens up the DNA. Its job is to break hydrogen bonds. There's another enzyme called topoisomerase that kind of holds the DNA as you are opening the bonds, because as you open it over here, it's going to make this part of it want to spin. It's going to make it twist. That's just like a practical physical aspect of like having a long stringy molecule. And so topoisomerase is going to stabilize as it's unwinding. Then we get to the star of the show, which is the polymerase. Polymerase in this case is DNA polymerase. Its job is to connect nucleotides together. Now, again, one small but important clarification is that technically the DNA polymerase is not matching things. When the enzyme shows up there, it makes it, my animation looks like it's grabbing a T and putting it there. But in reality, there's a lot of nucleotides that are fighting for that spot in the enzyme. And if a T fits, then there it goes. It's gonna fit, and it's gonna be able to get used to make the next part. But if an A were to try to go there, it would just bounce off, right? This isn't an active process. The real job that the enzyme does is when the second one shows up, it's going to make this bond, which of course my animation's a little bit off because whenever I switch things from one file to the next, it moves them a little bit. But this bond right here, that's the key. This is what we're going to make, right? We want to connect the nucleotides together. And when you do this, now it's moving even farther than I wanted it to, but you can see how it goes, right? It's gonna have to go from both sides because it can only move in one direction. In particular, it can only add right there, right? Enzymes can only do one job. They can make one bond in one place, and that's the bond that they make right there, right? They can only add to the 3' end. And so we would always be working our way these ways from 5 to 3, from 5 to 3. But then that does mean that we read from 3 to 5, and I'm pretty sure that's going to come up on my screen here in a second way. But again, that is an important detail that does get brought up a lot when they do ask questions about replication, if they ask them, right? So it's not the hard part isn't the base pairing rules, it's remembering this right there so that we have our, go back here, right? Our directions, right? The DNA is going 5 to 3 here, so we're going 5 to 3 there. Again, that's going to be important in a second here. After I mentioned that this is called semi-conservative replication, that we are always going to be making a copy of a template, right? And this is going to help us with the continuity of the information. So again, here we have an image of the replication bubble. And the next thing that's important is the idea that it is going in 2 different directions at the same time. But the DNA also moves in 2 different directions. So here I have my model of the helicase coming through, and it's going to be opening up the DNA right there, right? And there's the little yellow DNA polymerase. And sorry, it's a little bit jumpy on my animations, but the DNA polymerase up here is reading from 3 to 5, right? That's the normal way that we read from 3 to 5, which means there's the 5 here and here's the 3 right there, right? We're building from 5 to 3. But we can't do that here because that's not the right thing. The 3 to 5 here goes there, that direction, right? And so now we have a situation where we need a new enzyme that it has to go the opposite direction, right? And so the one polymerase can go with the helicase, and that's called the leading strand. Oh, and the other one has to go behind, and that's the lagging strand. There we go. I knew those were going to show up in a second. Right? So the top one has an easy job. It just goes in a straight line. But the one at the bottom, the lagging strand, has to be made in small sections because you have to expose one section of DNA, then add the enzyme, and it can work backwards towards what it already made. Then you get these things that are these little fragments. They're called Okazaki fragments, and we have to join them together using an enzyme called DNA ligase. And so that's the 4th enzyme that you might be asked about. We got helicase topoisomerase, polymerase, and now ligase. All those 4 together make this particular picture. Again, where you have things moving in different directions. This one's going that way, but this one is going this way in little fragments. And of course, the helicase is moving forward that direction the whole time, right? And so there's your DNA replication. The bubble will start to get copied in both directions until they fuse, and then you have your copies of your DNA. So that's DNA replication, but now we want to use the DNA to make proteins, and so we need RNA. We need, in particular, messenger RNA, but there's a couple different versions of RNA out there. All RNA are structurally the same. They're made out of the 4 nucleotides with, of course, uracil in the place of thymine. The important part here is the deoxyribose has no O right there, and ribose does have an O right there. And that it factors into its ability to make a helix and things like that. DNA makes a double helix while ribose will not. Single-stranded, U places T, the sugar. These are small details. Hopefully they wouldn't ask you about it. And of course you have your notes anyway, but these are what make RNA different than DNA. Now we want to use the DNA to make RNA. This is called transcription. We're going to make a temporary copy of only a small section that of course we call a gene. This one is a lot easier in the steps. We just need one enzyme called RNA polymerase. The only hard part is it needs to know where to go. This is a place called the promoter that's found in front of the gene. The promoter has sequences, for example, like a TATA box. This is just one common example of a sequence that is recognized by other molecules. The molecules are called transcription factors, and these are the things that bind, and if they all bind on the promoter at the same time, then you can go, right? So this is the starting line. This is a starting button, and the molecules need to know where to go, and that's what the TATA box and the promoter are all about. So you start at the start, and then you just open up the DNA. As you go, you do base pairing rules to make the strand, and of course you connect the nucleotides together into a polymer to make the final transcript, right? So here's my animation again. We don't have helicase, we just have the one enzyme that opens the DNA. Now this is an interesting small detail that one strand is called the template strand, because now if we match these letters, we would get slightly different information than if we matched up with those letters at the top. So we have to clarify which strand we're looking at, the coding versus template. The template strand is the one you match with. The coding is going to end up the same as the messenger RNA. So again, we just join our nucleotides together. The real key here is making that bond right there, right? The enzyme can only build to the 3' end, so we're still moving from 5 to 3, 5 there, 3 over there, and we're moving that way, right? And so we end up with an RNA that is the same here as there. Again, this is called the coding strand because it has the same code for the protein that we're looking at. So now we made RNA. This can go fly off to find a ribosome, except that we can't. In a eukaryote, we have to do other steps. We have to add a poly A tail in a 5' cap. We want to protect the ends of our molecule because the ends of molecules are where enzymes can attach. In particular, there's an enzyme that attaches right here, like a little Pac-Man, and starts to cut off the A's one at a time, right? And so you put a lot of A's at the end, and that gives Pac-Man something to do for a while, keeps them occupied, and your messenger RNA will stay available for longer. If you have a very short poly A tail, Pac-Man will cut these off real fast, eventually start eating the actual information, and then your messenger RNA isn't going to be used anymore because now it's broken. So it's kind of like a timer, and those things will protect the ends so that enzymes don't eat them. We also have parts of our DNA that used to be considered junk. They're not really junk, we're learning now, but these parts called introns that need to be cut out of the DNA, or the RNA rather, and then the exons get joined together. So this is like editing. We do call it messenger RNA editing or transcript editing, right? And so the benefit here would be that if a mutation happened in one of these spots, it wouldn't really matter because you're going to cut that part out anyway, and it gives protection to our really complicated genes that we want to preserve as best as we can. Prokaryotes don't have those because prokaryotes, their evolutionary strategy is kind of just like go as fast as you can, and if you make a lot of mistakes, whatever, there's a bajillion of us, right? So they don't protect their enzymes as much. They also don't have a nucleus to keep the ribosomes away long enough for the messenger RNA to be edited. So as soon as the messenger RNA comes out of the DNA or the RNA polymerase, the ribosomes just grab right on immediately. In our cells and eukaryotes, the ribosomes are outside the nucleus, so they can't just start to work on that messenger RNA right away. And that gives us time to edit and splice. Then once we make the splices, then we have this thing called mature messenger RNA, and now we're ready to go off to do translation. We need to translate it, right? It's in a code. We are going from a code of 3-letter groupings. Now, of course, those are called codons, if you remember. but the codons AUG, AAC, those are in code. That's where the word codon, of course, comes from. We need to translate that from codons into amino acids, right? And so for us humans, we use the codon charts. Remember, this can only be used on messenger RNA. It is not to be used on any of the DNA or the tRNAs that we'll talk about in a second, right? But with this chart, you can decode and know what the heck AUG and AAC are supposed to tell you, right? And so in the end, what's really happening, or the idea of what's happening, is that we take our DNA and we convert the information into RNA. The RNA gets translated into a sequence of amino acids, and as we know, the sequence of amino acids folds into the very particular shape based on its sequence, primary structure, which is the sequence, right? But in a cell, we need a mechanism. This arrow represents something that we need, and that something is called a transfer RNA, right? Again, same thing, made of A's, U's, T's, and not A's and T's. Sorry, A, U, G, and C. There we go. But this is the medium. This is the intermediary, right? In one side is amino acids. Sorry, nucleotides, and on the top are amino acids, right? So literally this guy is a translator. One half of it speaks DNA or RNA information, and the other side speaks amino acid, right? And so you have different codons with all the, or different anticodons to match, going too fast, to all the anticodons match up with the codons, right? So how do we know that MET goes in a certain spot? Because this little homing device will only match up with the complementary sequence of AUG, right? And so AUG will equal methionine, right? Or whatever these other ones are, because I don't memorize them necessarily. That is, of course, if we load them properly. I really hope they wouldn't add something like this into the condensed test, but because all actions in a cell need to be completed or carried out by enzymes, there's an enzyme that goes around and loads up the correct tRNA with the correct amino acid, so that we do in fact get the correct anticodon to go with the correct amino acid. If you were to get a mutation in this where somehow this enzyme is going to pick up the right tRNA, but instead of picking up that RNA or amino acid, it picks up a different one, and now you end up with the wrong amino acid onto the wrong anticodon, right? And this is going to mess up your protein. So that's just one small thing that comes up sometimes. Now we need a ribosome to help us do this. We need something to actually create the bonds between the amino acids. Ribosomes are kind of like gigantic enzymes. They are enzymes, or they are considered organelles with enzymatic functions. Is it an organelle or is it an enzyme? This is the third type of RNA that's come up now, ribosomal RNA, right? And so just to keep in mind that, like, for the tRNAs and the rRNAs, somewhere in your DNA is the sequence where you transcribe it, but instead of it going off to find a ribosome, it becomes a ribosome, or at least part of it. And then it goes in with the proteins to make this huge, crazy globular structure here that just looks like a big ball of yarn. But they usually gets visualize something like that, right? So here's my messenger RNA. Here's my ribosome that comes over. AUG is the start codon. We know that, right? So here's where we are initiating our translation. EP and A sites, just so that we have a way to talk about what's going on, right? This is the kind of stuff that you definitely don't want to memorize because it's already going to be on your notes. But you should remember the idea that the A is where things are going to enter, the P is where all the action is going to happen, and the E is where things are going to exit. So we need the correct tRNA to come up for this codon, right? Oop, going too fast. For that codon. Of course, this anticodon should have hopefully been loaded up with the correct amino acid. Now our job is to actually connect that, right? Again, remember that the ribosome isn't actually doing much here other than holding everything together. Up here is where the action's happening. This is the part that we're trying to make. So we're going to make the bond between the methionine and the serine. We are going to cut the bond to the tRNA, so the truck has dropped off its payload, its delivery. Then we can move forward one codon. I think it's going to get a little weird here. There we go. And we can do it again, right? The messenger RNA is calling for the tRNA that matches that, and so it should hopefully bring the right amino acid. We connect it to the chain, we disconnect the chain from the last tRNA, and then the empty one that was in the E site is now going to leave, right? And so we just keep going over this over and over again, where the ribosome lets the tRNA come in, it connects to the growing chain, it moves down one, the empty one just kind of ejects itself, honestly, just kind of falls out because there's no room for it. Eventually you get to the end where the ribosome is going to try to connect the chain there, but there's nothing to connect to. When it disconnects the tRNA, like it always does, there's now nothing holding that onto anything else other than holding them together to each other. And there's our polypeptide, right? And that goes off to fold into the shape that it needs to fold, assuming that you're doing this thousands and thousands of times, of course, because I'm not going to show that on my animation, right? And so in the end, we've taken our DNA, which is the mechanical storage device of all this information. We've opened the DNA, we have copied the DNA, we've used that copy to translate the nucleotide sequence into an amino acid sequence, and of course, amino acid sequence equals shape, and shape equals a higher order of information and function. Next video will be about how we turn on and off these, how do you control all this stuff. But for now, good luck with the quiz.